This meeting is now being recorded.
Well, good evening. Welcome to our EDS Awareness Educational Series webinar. My name is John Furman. Before we introduce our speaker this evening, we'd like to give you an overview of our free program for those attending for the first time. So we'll discuss who we are, what is our program, some upcoming events, introduce our speaker this evening, Marcus Bond, and then have a Q&A. So again, my name is John Furman. I manage the National EDS Awareness Program based in Cincinnati, Ohio. My daughter, Deanna, who is on the call, leads the Cleveland, Ohio EDS support group. She was diagnosed with hypermobile EDS in 2008. The same year, my wife, Carol, passed away with breast cancer. I was a caregiver for my wife, who struggled with undiagnosed EDS for over 30 years. We introduced our program at the 2012 EDNF conference to help EDSers form independent local support groups and spread EDS awareness in their community. We have been also a sponsor for a variety of conferences over the last 5 years. We started over 100 groups to date. Each group is given their own free website with a link from the directory map. We receive feedback that conferences provide valuable information and social opportunities that many cannot afford physically or financially to attend in person. So we decided to bring the speakers to you through the free EDS Awareness Educational Series. We meet every first and third Tuesday, typically at 7:30 p.m. Eastern Standard Time. All the programs are free. The meeting announcements and whenever possible, the webinar recordings will be posted on our website at edsawareness.com or chronicpainpartners.com for later replay. You can receive email announcements for future sessions by requesting the free guide on our site. Tonight's presentation is sponsored by Body Support Store, where you'll find over 250 products selected by EDSers for EDSers. The store is the only funding for this program and usually covers our monthly web fees. Please visit our store and check out the helpful products that we offer. Just a general disclaimer. This presentation contains general information about EDS. Members of the EDS community voluntarily participate in this program. The information is not advice. If you're having medical problems now, please call 911 or the emergency services at your location. Always consult your doctor before making any changes to your treatment.
We wanted to update you with some of the webinars that are coming up. The next one is on January 3rd. Jan Dammerholt will return for another webinar with us about physical therapy for Ehlers-Danlos. To view our upcoming speakers, always check the webinars page at edsawareness.com for the full schedule. For those attending live tonight, there will be an opportunity for Q&A after the presentation. Add your questions at any time by clicking the Q&A icon at the top of your screen. After typing your question, click the orange button to submit. Tonight's presenter is Marcus Frederick Bond, who will provide an introduction to tenascin X and Ehlers-Danlos syndrome. Marcus is currently a postdoctoral scholar in the Department of Pharmaceutical Chemistry at UCSF in San Francisco, California. Marcus has a special interest in Ehlers-Danlos syndrome, and he is also currently interested in identifying novel therapeutic targets for antiviral strategies at the host pathogen interface. And his education includes, he graduated from the University of Massachusetts Medical School with a PhD in natural sciences, working in structural biology. His graduate and undergraduate studies were at the University of Erlangen-Nuremberg in Germany, where he graduated from the master's program in molecular and cellular biology. So it is a great pleasure to introduce Marcus, and we are looking forward to learning so much about tenascin X, and we're going to go ahead and load your slides for your presentation on the screen. And I'm going to go ahead and turn over the microphone to you now, Marcus. Welcome. Thank you for joining us tonight.
Perfect. Thank you, Deanna. Yeah, and thanks, John. So without your great organization, obviously this series wouldn't be happening, and it's a great honor to be a speaker among so many people who have shared deep and profound insights, and that I get a chance to show the little contribution I did and why I think this is an important topic for the community. So I will, see if I can, so I'm wanting to give a general overview about the structure of the extracellular matrix. Then tenascin acts as an organizer of connective tissue within the context of the extracellular matrix. Then how that leads to EDS types, where tenascin acts is involved because of the role it's playing, and then the recent advances in research on this topic. So as you might have noticed from the introduction, I'm not a medical professional. I'm a biomedical scientist at the University of California, San Francisco. And I've been doing protein biochemistry for the past 10 years, since I joined the lab as an undergrad. This is what I've been doing all day, every day. And what I mostly worked on in the past was at the host-virus interface of pathogens. But for things that I will get into later probably, I developed a huge personal interest in Ehlers-Danlos syndrome, and realized that as a scientist, there are skills I can bring to the table and try to also contribute to it and help our understanding and lead, if not to useful therapeutic or diagnostic options, then at some point at least to insights and knowledge. Because the lab I'm currently working in, we're interested in developing novel tools for imaging and diagnostics, and so this is more or less tangential to that. So yeah, and I will probably try to get that perspective and then explain why it is so hard to develop diagnostics or therapeutics for in general for connective tissue disorders and especially EDS. So the extracellular matrix is exactly where the problem lies. We all know our body is organized by cells that have functions, and where the action is where metabolism happens, where we build things and metabolize things, and where thinking happens. And between those cells is a matrix, so that acts as a connective layer. And you never see that in images of tissue or something. It's usually it's usually treated as just a barrier or a glue between those, but there's a lot of action happening there, and we will look into that because the extracellular matrix has to provide tissue infrastructure for our organs and how metabolize chemicals, but also electricity that gets distributed, for example, in neural contact, that it's not leaking. So these properties are provided by the matrix. That it provides a linkage that's mechanically and biochemically between cells. And there are lots of components, and it's a ridiculously highly organized organ in our body that pervades every tissue that we have. Then for these reasons, it's obvious that anomalies in the connective tissue can have an effect at health. And I just grouped a few here so we all know. Heritable is, for example, EDS, or also Marfan. There are some autoimmune ones like SLE and arthritis, and there is the odd outlier that's scurvy, which is actually interesting because scurvy is one where it is a metabolite. So that's what we know so much about it. We know you can take vitamin C, and scurvy is you're deficient in synthesis of collagen, and those things are much easier to study and much easier to approach. and instead of the other two options. Because what we know, what all the other ones have in common, if you know a person with rheumatoid arthritis or SLE or EDS, a big thing is those are generally not really treatable or curable diseases. Like you can alleviate the pain that arthritis does. But as if you're a rheumatologist, then you get to know your patients really well because you will have them for life, and it's not changing. And the same is for SLE. I worked on SLE in the past too, so it's kind of odd that it basically came back in this pool for me, where you develop autoantibodies against your own tissue components, and also there nobody really understands what the mechanism behind it is or what a valid therapeutic approach is. And the same goes, of course, for the heritable ones, because we can identify that there is a genetic cause. We can sometimes nail it down to individual residues on a protein chain, which we'll do later in this talk. But that still leaves not really a hook to do something about that, to affect it and provide some options to treat it, or as a lot of people who suffer from EDS know, at least an easy option to get a diagnosis really quick. Because a lot of the SLE is, for example, would be easier to diagnose because you have autoantibodies, so you can make an assay against a protein that shouldn't exist. But in Ehlers-Danlos syndrome, you do not have that luxury. There is no component per se lacking. There's this general phenotype of tissue disorganization, and the causes for that can be varied. So in that, it's more of a roof diagnosis if you get called out for Ehlers-Danlos syndrome, because there could be so many underlying causes that affect it. Tenascin X is a connective tissue organizer, and I put that in quotes because that shows that tells nothing basically. So you know that it obviously plays an important role about how a tissue is structured, but you don't really know what it's doing. So the role of an organizer is very hard to describe. It's like in real life. It's easy to describe a job that revolves putting, for example, a lid on a jar. This is what would you be doing. But if you're managing a company, it's much more complicated, and the same here. Tenascin X plays a management role in the connective tissue, and it's a huge protein. So this is the next line, shows how it's structured. So it's 4,200 amino acids, which are the building blocks of individual chemicals linked together in a protein. And it has 19 EGF-like domains, so which is a description for a domain that's first described in epithelial growth factor. Then it has 30 fibronectin type 3 domains, and then it has one little terminal fibrinogen domain. And just for the scale of that, I'll put a simple chemical reaction that most people will know because they've experienced it, and that's the metabolism of alcohol. So when you drink alcohol, then at first the alcohol dehydrogenase will come in and turn it into acetaldehyde. So that's the substance that gives the bad feeling on the next day, if it lingers, because that one then gets slowly metabolized by aldehyde dehydrogenase to acetate, at which point it's not an issue anymore. So if you see, the green boxes correspond to the domain structure of those enzymes. So if you compare those to the size of tenascin, those are tiny. So which makes it easier to handle for studies. If you study something that has a lot of parts that are interlinked, that's harder than if it only has two little domains like ADH, and the same for ALDH. And also they're enzymes, so they have like an intermediate product. If you see that a person has a defect in ALDH, you will immediately notice, because you can monitor the buildup of acetaldehyde, and you will see that there's less acetate. You can even just make those enzymes and do it in vitro and just study what's happening and see, what does this mutant specifically change? Does it change how ALDH binds to acetaldehyde, or does it change how it releases it to acetate, or does it change how the conversion between those two things happens? So this is a very easy to study process, and so we know a lot about it. We know a lot about metabolic proteins because those provide access points where you can insert an experiment. And then also in the end, it provides, for example, a druggable point. If you would, for whatever reason, not want that somebody could metabolize alcohol, you could just throw in a little chemical compound that blocks ADH. and then this whole process wouldn't happen anymore. And this is one of the reasons why I worked on viruses, because this is how antivirals work. You just block something in a pathway that's really important, and then that's it. But with tenascin, that doesn't have a function per se, that it doesn't convert something. It doesn't have some measurable function per se. The only measurable function is more like in that bridge collapse pictures that I picked. So you know when it's working because you have your bridge. You can pass over it and it's behaving nicely. And then the only time you notice something is off is when the thing on the right happens, when it's collapsing and not working. And there's if you set up your bridge wrong, there are few things you can do after the fact. And the same happens with hereditary diseases, because if every single copy in your body is making something wrong with tenascin X, then for one thing, we have nothing to measure, since it doesn't produce anything that would be detectable for us, nor could you do something after the fact. By the time your tissue is built, it's done. There is nothing you cannot go back in and re-engineer. And that's why this is such a challenging target. And I got so excited about the bridge pictures, so I had to dig out a few more pictures about what tenascin X function implies. One is the collagen fiber architecture. In the top left, so the red little blobs in there, those would be the bridging tenascin molecules. So you would see in tissue, you would see tenascin between the collagen fibers, and it would be attached and doing something. And you would know that something is wrong if it's absent. There are tissue phenotypes. If you make no tenascin, then you will see tangled fibrils, like in the picture on the right. And they will affect elasticity. Like if you would have, like with the fabric, for example, below it, you can have failure in elastic fiber properties. You notice that depending, you will probably have noticed that if you wear fabric, for example, that some parts would need to have different elastic properties than others. That's why they rip easier or tear and get used down. And in your body, you don't have the luxury to just make a uniform fabric and then say, oh, we can have rupture. So you need a very precise distribution of these properties. And if this is not successfully made, you will see the same thing. You will see ruptured fibers. And this is what people see in the biopsies of it. And what could be almost more importantly, but it's even harder to study, is a failed mechanosensation in a fibrous network, as see with this poor spider here in the top right that has the same issue basically. So this net does not provide what a spider does, because a spider web has, besides, I thought there was a good analogy here, since it provides more than just the net property. It's not just the superstructure of the net, but spider sits on it and uses tension and behavior of the strings in order to figure out what's happening in the net. And the same thing is happening in your body. Your cells in your body, they cannot see what's happening around them. So they're very well tuned to be sensitive to changes in the direct surroundings. So signals propagate through the networks that connect cells. There even death signals can be in part contributed to that signaling network. And failures there then mean that your cell becomes blind to its surroundings. It doesn't know what to do. Can easily be manipulated by factors it should not care about as soon as it loses connection to its surroundings. And that basically, so those defects link tenascin then to the types of EDS that it affects. So we know there is a type of tenascin deficiency, so it means there is none. So the cells do not make tenascin X. But that's measured through serum levels of tenascin X. So there's also, if you have a protein that has no measurable function, as I mentioned before, like an enzyme, then it's also very hard to know that it's not there, because you have to measure it directly. But since your body has so many different compartments in terms of organs and tissue properties, sometimes it's really hard to do that, because you cannot just throw a reagent on it and it turns blue if it's there or something. You have to measure serum levels of it, which is is a dicey thing. So these people who have a deficiency, there is no measurable serum level of tenascin X. And they have a skin hypertensibility, hypermobility, bruising without trophic scars, and neuromuscular issues. And that opened, this tab opened the door for experimental research, for example, because a deficiency you can make. This is what you do in model organisms. So you can take, for example, a mouse and just knock out tenascin X in the embryo, and then that mouse will not make tenascin X. And that has been done in 2002. And there's a double mutant because you usually carry two alleles of the same gene. And that one shows the same phenotype. It was a big breakthrough because it showed that tenascin X on its own can explain a lot of the skin hyperextensibility that's observed in patients with the mouse model. So that's really good because that nailed down that that tenascin X isn't a proxy for something, that it's actually a cause. And the neuromuscular issues there wasn't a good model so far to study that, but I know there were some very few exciting experiments on chicken. Chicken don't make tenascin X, but they make a sister protein of it, and that one can be studied to localize to the neuromuscular junction. So mentioned that cell contacts are made to the extracellular membrane, and so that gives a lead that there's a tenascin role involved there, but that's also hard to study. And there's the haploinsufficiency, which means that people make precisely half the amount. As I mentioned, you carry two alleles of tenascin X. You get one from your father and one from your mother. And if one of those is defect, then you only have half left. So once you transcribe and make protein, you notice that you have a dose effect, basically. You just don't have enough. So the symptoms those people have are similar, as described, similar to hypermobile EDS types. There is not much of a skin phenotype compared to the complete deficiency, which also makes sense, since that is a much milder form that happens. Then there are missense mutations. So EDS phenotype without any sense of deficiency or haploinsufficiency. So it means you can measure protein level of tenascin X, but people still have problems, like skin hyperextensibility and the general hypermobility. And this is really hard because to do that, you have to collect a lot of data on individual mutants. And so that means out of the few people who have mutations, you need to cluster those in order to get some provocative data. And and that means there's a lot of data collection necessary. And we will talk about that because this is where I came into play to do something. And that happened in combination with the last tab, with CAH-X. That's a special subtype of congenital adrenal hyperplasia, which is an endocrinal disease. And that happens, those people have mutations that lock down their CYP21B gene. CYP21B is adjacent to TNXP. So there is a 7% opportunity once you have your CYP21B defect is messing up the TNXP right next to it, and you get the pleasure to have both CAH and EDS at the same time. So you basically get a haploinsufficiency because then one of your tenascin X is down, and at the same time you have to deal with the endocrine issues of CAH. And the next fun thing happens once you have CAH-X, you have obviously only one TNXP allele left. On this one allele, if you get TNXP missense mutations, then you get your own very unique EDS type, basically. But it also allows to study the effect of a missense mutation really well, because with the two alleles that you carry, it's hard to tease out single mutational changes, since it's usually compensated by the other copy to some extent. So whereas with CAH-X patients, this is doable since they carry both. And that led to basically the project, how we call my project, and that was part of a larger project at the NIH, that's bridging complex genetic heterogeneity. So the heterogeneous population of people with CAH-X and the spectrum of clinical phenotype that they exhibit using protein structures. because that is my area of expertise. And on the example of TNXP. So just a little bit more for the CHX background. It's hormone deficiency in combination with EDS, and CHX is also so that is an enzyme deficiency, as I mentioned in the beginning. And in this case, this is the 21-hydroxylase, which is encoded by CYP21A2. And what it does is these steroidal hormones, they need to be hydroxylated at position 21, which is where the arrows are pointing. If you're not able to, hydroxylating just means adding an OH group to it. If you're not able to add an OH group to that thing, then you suddenly have an insane discrepancy in your mineralocorticoid metabolism, your adrenocorticoids. You get insane issues, basically with your sexual steroids, and that's just for a little tiny change, but it's measurable. It's a huge clinical outcome for the people who have that. And what happens for people with CHX is what you see on the right. You have in blue, you have the exon, so the coding region for CYP21A2, and in red you have the coding region exons for TNXP. So they lie adjacent. So what happens if you look below it? You see there are a few gray boxes that have the outlines in blue for CYP21 and in red for TNXP. So the locus in which these genes lie is a very fragile thing. So there's a lot of things that can go wrong, and this is one of the reasons, because they're pseudogenes. So genes that are derived through copying TNXP and copying CYP21A, but that became defunct over evolutionary timescale. So they cannot be expressed, they do not make a functional protein, they just lie there. Now, during meiosis, so the process when the chromosomes match up and segregation happens for diversity, so the sister chromatids come together, and what you can have is something that's called unequal crossing over. So this is with the little blue and red X that's happening in panel B, where you can have your working copy of CYP21A aligning with a pseudogene, the gray box with a blue outline, and the same for the TNXB and its pseudogene that's called TNXA. And because the sequence is really similar, as you saw, especially TNXB has a lot of very similar domains. It has 19 domains of one type, 30 domains of another type. So those can be lined up, and then you can switch out through a genetic mechanism one with the other, and put basically what happens in panel C. Then you can put your TNXB behind dysfunctional pseudogenes of CYP21A and TNXA. And then it's not being transcribed anymore. That's the mechanism that happens. And at that point, then you lose both. So you will not have the 21-hydroxylase anymore, and you will not have TNXB. And on top of that, things that happen all the time is point mutation. So usually, in this case, in this study, the patients, they had inherited those point mutations, but they can also arise spontaneously. And that was what I did my PhD work on, was on an enzyme that introduces point mutations, specifically in the DNA. We do that actually on purpose sometimes. So we introduce point mutations, for example, when we generate antibodies. So our antibody diversity, the fact that we can recognize unknown pathogens, is in part built on the ability to introduce point mutations, in this case, in recognition loops of antibodies. And the same enzyme family is also capable to introduce point mutations in the regular genome. And one reason might be to generate diversity during spermatogenesis, for example. But it can also be completely dysregulated as in breast cancer, which is from the same enzyme. And point mutations, there's three types, and what we care here about is one that changes the sense of the coding triplet. So the DNA bases, as a lot of you may know, they encode in triplets. So three bases make a codon that encodes for an amino acid. For example, here CAT gets read out into the amino acid histidine. And if you replace a single nucleotide, then you can change the meaning of that. And you can, for example, replace a histidine, which is pretty bulky positively charged amino acid, for example, the proline, which is a sterically extremely restricted one. If it happens to be in a helix, for example, it will break that secondary structure, and I'll show you what that actually means. And that might lead to a malfunction in protein, just one single change. If you remember, from the previous slide, the size of tenascin X, since we mentioned that, right? We have 4,200 amino acids. We have 3 times the number of bases then that encode this protein. And if you get one wrong at the wrong point, then there is a good chance that you mess up what's happening in the end. And the trick is to know at which point that's happening. Because you're able to carry mutations, of course, that do nothing, that do not change the sense in the first place, that don't fall in a coding region, or that happen to make a change where it doesn't matter, or exchange an amino acid for one that makes no difference. And here is the group of Deborah Merkel at the NIH. They had a group of sample of patients with CHX, and they sequenced their the remaining TNXB locus to find point mutations that might explain differences in tissue phenotype. In this case, there were 3 patients in this sample, and as you can see from the pictures, they all have very clear EDS, skin phenotype, and hypermobility. And what I'm going to pull out here is just the most protein biochemically most interesting example, and that's what's circled in red. That's a patient who had a C to G mutation at base 12,174. So this single change in one out of more than 12,000 bases happens at the very terminus of tenascin X, as you see where the arrow is pointing to the right in the fibrinogen domain. So below, I color match basically how you have to imagine how those domains look. So the EDF domains are from a solenoid, they're short, the FN3 are a bit bigger, and the fibrinogen is the largest one. So this single base change in this domain on the right, that's in red, at the position where the arrow is pointing in the domain, replaces the amino acid cysteine with the amino acid tryptophan. So this turns out to be a pretty dramatic change because tenascin X uses cysteine to form bridges within the molecule and between molecules. So a lot of proteins are able to interact with other proteins and to form solid links, and the cysteine amino acid is the one that's responsible for it. Whereas tryptophan is a very bulky residue that basically in this case just disrupts it. And and this is basically the reason why I got looking at this type of thing was what got me into this collaboration because my wife is an EDS patient, and so far there has not been any real cause found for why she has EDS. And but she carries a point mutation on her tenascin X, and of course it wasn't characterized because it's hardly characterized for anybody. So I did calculations and we reached out to the NIH and said, well, is this something? And said, well, if you do these things, you could do it with us too. And so this is a great opportunity then to help with that. As you notice here, for example, so this patient only had one mutation and had a pretty pronounced phenotype. The other 2, they had a cluster. So you have suddenly you can have 3 or 4 more changes in the same domain that swap out a residue for another one, and that will then cause some unknown type. But here, for example, with all of these, what all these 3 had in common is that you could say that that the effect it has is destabilize the domain, so it unfolds easier. So it becomes too flexible. It's not structured anymore, and that's why I figured this was a really important insight, because since tenascin X has not a measurable function per se, except when you observe the tissue phenotype and defect, knowing that just chemical stability of an underlying domain can be causative for difference in tissue phenotype, that gives kind of an angle that can be approached, at least theoretically, in experiments. And that is also what's happening in the field. So people did in the past, for example, measure just the stability of domains just by pulling at them. So you can do that with optical tweezers, it's called. So you can just basically take out a protein domain, put it between those, and then measure how much energy it takes to rip it apart. And that was done also with the TNX, tenascin X at some point, but without having a good handle on what to try. So which mutations make sense, which do we know are disruptive mechanically versus which ones could lead to other defects in the organization with interaction with collagen, for example. It's very hard to do these experiments, and this is also what happened. So they didn't see much in the past. So a change of I think it was 20%, and the error on it was 15%. So barely noticeable difference. And and this kind of what Dr. Merck is doing, the collecting of patients, just alone the collecting of patient data of these mutations will really pay off in the long run, because we will be able to generate a pool of mutations that can be clustered and see which ones go together and can lead to which differences in the tissue phenotype. And that's what it looks in the big picture of a story, basically. So you can trace in the family how a similar defect allele could propagate and then get inherited and lead to what it does, because in this case it gets found since the patient was haploinsufficient because of CHX. And you can see in the panel A on the right, you can, for example, see how the tissue phenotypes are for elastin, how a normal tissue looks versus the defects in the 3 patients and the fibrillar organization. And a lot of these changes are pretty dramatic. And the difference between the patients comes from the difference in mutations they carry on their one single tenascin X allele. And so in the long run, there might be a chance to characterize these mutants and then use genetic data from whole genome sequencing or something to be able to tell people, oh, see what you're having might be because your mutant of tenascin X can cause these things. And I think that would already be really helpful. And yes, and this basically already concludes my little talk. So if you want to, if after today, if you want to shoot me a message or have a question, feel free to send it to my work email here. If you want to see what I work on in general, or the paper I just mentioned about with Dr. Merck's group, there you can see those on my UCSF website. And with that.
Okay, and we can
yeah.
we can go ahead to some questions if you don't mind.
Yes.
I think there may have been 1 or 2 questions submitted to you ahead of time.
Yeah,
do you have those?
Yeah, there was one ahead of time, and that was actually a really good question. That was I think the big gist of it was the vascular phenotype and aging, and the only thing that was found was a mutation in tenascin X. And if there's a correlation, just double check so I'm not guessing a question that I like. Yeah, with the aging and the vascular phenotype, that it can be correlated and it can show up later. And that's a really, really good point, and that is a part of this aging, because we all know what there is for us ourselves. If we just look at old photographs, there is a visible change in tissue phenotype when we age. So we all know that we lose joint elasticity when we age. If you compare a toddler with a 70-year-old person. But the fun thing is, I'm not a medical person, but what I read and heard is that a lot of the cases you don't see a lot of so the tissue changes between a young and an old person aren't that dramatic, but you know that the effect in the large scale is really dramatic. And if you think about how your collagen elasticity, for example, changes during aging, and there's a lot of cosmetic companies that try to remind you daily of that's happening, and that's a bad thing. And the tenascin X is your organizer of that. And suddenly the material it has to work with gets worse too, because it gets damaged by sunlight. You cannot restore it as quick as you could 50 years ago. Then of course that can aggregate. And the science of aging on its own is a very fascinating and huge topic, and it's awfully complicated. And this does not help with that. So I believe there is also an aging component about how that can affect it. And then they can also be helpful because we all know that joints get stiffer over time, and I think some was described that it can actually help with some of the EDS complications in the long run. And another question was if there's a genetic testing service such as 23andMe that can identify the mutation, and the answer to that is yes. So we had that when I did a Q&A on Facebook. A lot of people asked about the 23andMe and sequencing data, or what type of sequencing they should use, and that's a really up and coming technology. That's something that will happen on a routine basis as soon as it gets even cheaper. And so these services do that, especially 23andMe. They do 50 reads over the same gene. They can be thrown off by the pseudo gene. So that can happen. So what happened to some people is they told in their readout that their tenascin X locus couldn't be read because of the pseudo gene. That's precisely the problem. So the readout tools that 23andMe uses have the same problems that cause all that headache for the CHX patients, basically, because of the crossing over. And so also their probes can align to the wrong protein and then cannot discriminate anymore what they sequence, if it's the non-usable copy or if it's the TNXB gene. But otherwise, yes, you can use that, and you can see what your mutant is, and theoretically it's possible to just aggregate that and then see what people were having, and then do what I did, the biochemical, it's computational analysis of biochemical properties. And at some point that would make a lot of sense to do that on a larger scale. Then how does a person with testing for tenascin X? What geneticists suspect. So I'm not a medical person, so I'm not familiar with the workflow. But I noticed there's this triad of evidence for persons of EDS, right? There's the Baton score, so people measure your angles and mobility, and they see, oh, there's too much of that, your EDS. But that gets trumped, of course, by the tissue phenotype. So if people see that your collagen fibrils are disorganized, then that's a pretty good lead that something is wrong. And then the backseat to that is the genetic testing. So usually people start doing genetic testing if they cannot find anything wrong, but the person has issues, right? If there's no other clear thing missing in the whole equation, then usually the sequencing is done, and then, oh yeah, there is something in tenascin X, but that's usually not very, very clear if that's conclusive, which is exactly the problem that my wife was having. So she had a clear EDS clinical appearance, but then everything was negative until the sequencing was done, and then it looked like there is a good link that this might be the cause for it. But again, there are no certain symptoms that, besides the general EDS phenotype, that could bring one to check tenascin X. And tenascin X, along with collagen and those genes, those are candidates. But one of them might be causative for the EDS phenotype. The past and future research. So the research on tenascin X goes back to 1997, I think. This was when for the first time was described that then could be causative for EDS phenotypes. But what was mostly done, so the big landmark per se was the mouse model, translated to mouse model, and then a few attempted biophysical studies. But what would need to happen is to basically fill the gaps. So to go from genetic findings to the clinical phenotype. This is where the huge disconnect is, and this is what's the case for all connective tissue diseases. Basically, it's not just an EDS problem. It's the same problem with also I would say it's the same problem with even arthritis and just heritable ones like Marfan. But there isn't a clear clear path that leads from one to the other. For example, if you could say that a cysteine residue in the fibrinogen domain makes it a tryptophan, and because of that it cannot trimerize on the collagen, and because of that the collagen fibers get not separated by 50 nanometers but by 80 nanometers, and then because of that the elasticity increases by a certain value, and this would be really useful and has to happen at some point, but it's hard because there's no clear-cut way even how to develop the methods to do that. And aside from that, it's also the problem with suitable animal models. As I mentioned, there's the mouse and there were some chickens, but especially if it's in regards to understanding the neuromuscular junction, there isn't really much to go about, since dealing with neural cells or neural tissue is incredibly hard to begin with. It grows really slow and is finicky, and finding basically then even establishing a tissue model is very difficult. But people have done that, for example, in the chicken model, and I think that's the gist of what's happening. There is, surprisingly, considering so the connective tissue disorders, the hereditary ones, they do not have the same glory attached in research, political, as for example Alzheimer's or cancer, even though they affect a huge amount of people, and there should be a huge interest by, I think there could be even a huge interest by the cosmetics industry if we're able to figure out how to remodel tissue by manipulating, for example, the organizer behind collagen. You could tweak tenascin X to do what you want. You could probably do a lot of things besides just helping people who are sick. So you could probably get very rich on that IL-2. The symptoms that lead to testing for tenascin-related EDS. Well, usually it's the same as for EDS. If people have not found a cause why you are having an EDS tissue phenotype or why you have EDS, then sequencing, just doing a sequencing run is a good idea, because very likely it has a genetic cause, and the whole genome sequencing will show something if a candidate lights up, and tenascin X is one. But of course it doesn't mean it has to be. So it doesn't have to be that if there's nothing else found, then you need to have a tenascin X mutant. You can still have EDS and your tenascin X looks just fine. There are probably other candidates in the networks of interactions that might play a huge role in getting to the same outcome. At UCSF, doctors that work with EDS patients, specifically in neurology, that is a question that we dealt with for a long time. Since my wife is an EDS patient and I work at UCSF, we would have been insanely happy if there was something we could do here. There are people here who are tangentially interested in it, as for example, as happened with the mouse paper, but that was 14 years ago. It's usually in the context of that sounds really sad, and is that it's usually in the context of more fundable projects, and that's the same at the NIH too, right? So their EDS we did in the context of people who had CAH, because this is what endocrinology was supposed to do, working with CAH, and then you could also then work on tenascin X in the context of that. And same here, there are people who were interested in the role of tenascin X because of, for example, lung development. So if you're already working on lungs, then you can work a bit on EDS if you can tie it to lung development, and that's how science is organized. That unless there's a lot of interest behind a project, you have to find a way how it explains something else. And so sadly I cannot say that there are doctors that work with EDS patients. But if anybody would run into some, then feel free to tell me about it, because I would be very interested to sit down and see if there is something we can do here. any research that could be promising. As I mentioned, there is a lot to do, but there's just not there's not a clear-cut path. It's not just that it's not easy to fund, but usually if you apply for funding, you need a path in front of you, what you think, what you can do, and what it leads out, and a clear timeline in which to achieve that. And with the entirety of extracellular matrix proteins, that's very hard. Since we don't even have, there would need to be some method development in the first place. You need a tissue that you can grow in the lab. You need a tissue model. You need cell models that have a high throughput. You cannot, for example, make a mouse for each of the mutants that is there. That's just not feasible. And even then, you wouldn't know if it translates into humans. So having human tissue culture is really important. And and hypothesis, the RCCXY, that's a good question. I read that website. There are some things that are easier to read than others. Per se, this module that exists and has been known for a very long time, as I mentioned, it's a fragile locus. There are some loci in the genome that are just known to be prone for things breaking and misaligning because of what I mentioned. If you have a huge number of pseudogenes, that can happen, or if you have a large copy number of very similar genes next to each other. And where CYP21A is located, there's part of the human complement system. It's called C4. So the complements are numbered, and the goal is they're part of like a primary immune defense line. They make holes in organisms that we don't want to be infected with, and in the complement cascade. And they also play, as almost everything else in our body, a role in development. And of course, it's common because they lie next to each other. So none of these things is per se new. But then linking already tennyson X mutants with specific EDS outcomes, as I just showed, is hard. And and most of it is speculation, because even if I say that there's a cysteine to tryptophan that's disruptive, that isn't measured, right? That's predicted. So that would make sense in the context of everything else we know about the protein and how proteins work. And going beyond that is really hard. So I've never worked on something that's a clear cause for a mental illness, basically, in what I'm doing, because it's a very hard topic, right? So, and to discriminate that. And also a lot of mental illnesses become suddenly non-mental illnesses as soon as there is a biochemical cause for it. For example, Alzheimer. You wouldn't per se say a person with Alzheimer has a mental illness if you know that there's prion protein like built up if you see the clotting. So so the Ankerg is saying probably common. There are mutations all over the place that are common. You have 200 mutations that neither of your parents have, for example, because that's just what happens during how we propagate as humans. So you get through, part of it will be at some point will be understood are ApoB mediated, but through mutational processes you get your own little chunk of newness that nobody around you has. And most of it is nothing. Most of it will just give you character and, for example, change your eye color or make you wear glasses, even though nobody in your family does. But some of it will have a larger effect. And how common those are, we would need just a vast body of sequencing data, which we will likely have just in a few years, as soon as the genetic tools that are used now mostly in the clinic and in the lab will make its way into a physician's office. And if sequencing costs $100, then it will be something that's probably done a lot. And we will have that at some point.
I think that may have been the last question.
I think that's it. Yeah.
Last call. Okay, wonderful. You made it through a whole bunch of them. Thank you so much.
That was good. Yeah.
Oh, there's so much information that you have for us, and it's all brand new to
I'm sorry.
us, and it's so interesting. It's so interesting. We've got to learn more about this. And I so appreciate that you're so interested in trying to do future research. And I hope that people who are listening to this will keep that in mind. And if we can, get some opportunities to do some more research on this, it would be quite helpful. Is there anything else that you'd like to say in closing? I think we're out of questions, so we're going to go ahead and wrap up for the evening. I think everybody learned a lot, and I really appreciate all your visuals to help us try to understand the scientific aspect of it and what's going on with the structure and with the mechanical properties going on with the defect.
Yeah, no, I think I think I got it all through, and and I don't know, as a closing word, it's just whoever has to deal with any of this, just hang in there, so you're not forgotten. It might be hard to find by the right people, and it might be hard for you to find the right people, but it's going on, and there's a lot of good people working left and right to see if they can make a difference.
Oh, thank you. Thank you for your compassion. And it definitely hits home for you, I'm sure, with your family affected by it. Okay. Well, again, we appreciate everybody that's attending live is thanking you now here online, and we appreciate you sharing all your expertise with us tonight. We are going to remind everyone that our next webinar is going to be on January 3rd, so don't miss that. That will also be at 7:30 p.m. Eastern Time, and we will have the topic of physical therapy. This is the second presentation by Jan Damerholt, who's a doctorate in physical therapy. And you can, of course, check our website for also future presentations, which we hold typically twice a month on the first and the third Tuesday evening of each month. And I wanted to go ahead and turn it over to John to close out the meeting for us now.
Yeah, again, Marcus Vaughn, we appreciate your participating in our educational series, and we're learning a lot of very interesting topics, and it's exciting to hear that there's research being done in a variety of areas that are eventually going to help our EDS patients, and you obviously have a passion, as I did, because your wife has EDS. So I want to wrap things up. I want to remind people that these programs are free, but for those in the U.S., we do have a little store that helps to support this program. It's called Body Support Store. So please go out and check out some of the products that we have, that we offer. And again, those that are on the call, have a pleasant holiday, Happy New Year, and Merry Christmas. And we're going to wrap up for this evening. So again, thank you for joining us, and have a good evening. Good night.
Thanks, Marcus. Thank you, everyone, for joining us. Good night.